Yeah, so this is kind of like a brain restoration, and I don't know if people have thought about this or talked about this a lot. I know that we've talked a little bit about it in fellowship, but we want to be able to present. So I just want to review here, right? And this is what I do for the students. We always go through that sleep is not monolithic. It has different voltages. It has different things. It has all sorts of waves. It has spindles. It has K complexes. It has delta sleep. It has REM sleep. So the brain manifests itself with electrical activity that changes over time. And what it's doing is often not very evident by watching someone. Although I must say that Sushil, in the recent chest journal, there's a part 2 on Arabian medicine that talks about sleep that's really quite interesting that we should put around. It says that there are 3 types of sleep that were determined on the basis of breathing and pulse. So we know aging changes the sleep, and this is 5 years of age, 85 years of age. And the REM sleep over that course of the time is fairly stable. Slow wave sleep diminishes, stage 1 and 2 sort of vary, and wake after sleep onset increases. Now these we're going to go on to suggest that these things are quite variable among individuals. But as for a sort of a general species approach, now remember that 80,000 years ago, mean life expectancy was somewhere around 55, but now ours is up there above 75. So that's what we need to sort of think about. And that the depth of sleep, 3 years, 23 years, and 75 years. The slow wave sleep, or the pattern of transitions, which occur in every one of these ages, is a little bit different. And in particular, what is termed slow wave sleep, or the depth of slow wave sleep, as detected from electrodes placed on the scalp, on the brain surface, changes. And some of this is due to the thickness or of the skull, as well as the size of the brain, and it's whether it's away from the skull. But there's a reduction in slow waves that occur. So the question really becomes, why is there this sleep, and what does it do, and how does it function? And what's the impact and need for these state changes, and what is the impact of the changes in slow wave sleep that occur with aging? So 3 objectives. One is glymphatics, and glymphatics is a term used to describe how the brain functions to get rid of waste. Second is neuroplasticity and adaptation. Can sleep and sleep states alter waking systems? And the third is something that actually we worked on for some period of time on ontogeny. As does sleep play a role in the development of the brain and its functions? So again, here are these different EEG patterns that you can measure when the person is going to sleep, and then goes into these different sleep. So glymphatics is brain maintenance. So how does the brain take out the trash? So medical school, they talked about 2 barriers. One was the blood-brain barrier in this region, in which there was a endothelial cell that was around the capillary. There's a basal lamina, and if there are different podocytes that are around these capillaries. And the second is this blood CSF barrier, and a couple of different parts of the brain that could be leaky. And this choroid plexus and epithelial cell impact junctions and interstitial fluid. So that's sort of what I learned in medical school, and which is often often brought up in mechanisms of things like brain inflammation. But and now on the right are really the anatomic sort of representation of how arteries go into the brain. And they come through the pia, they come through that area, and then how do the veins go out of the brain? And of these spaces in between the artery and the vein, and what they represent in terms of this potential space and what's going on in between this artery and the vein, and this has important trophic factors for the cortex. So what are these lymphatics? Well, it's analogous to the lymphatic system that is found in the rest of your body. Lymphatic vessels clear interstitial fluid of waste. And when we generally think of the lymphatics and what do they do, we think of them as an immune function. We think them as a clearinghouse for getting rid of proteins, and we don't necessarily, they're large, and particularly in things like the lung, you think of it as going up through the pleural surface and then being able to be dumped back into the to the veins. And then if you have lymphatics that are in the legs, you know, if they get interrupted, you have lots of edema and you have lots of problems in there in terms of drainage of lymph fluid, which is can be clear, but it's often filled with lymphocytes. It's filled with immune function cells. It's filled with sort of misfolded proteins. So the brain does not have a lymphatic system. It has what's called the glymphatic system. And it's really convective flow between cerebral fluid entering through arteries and interstitial fluid exiting through the veins that removes the biomedical waste from the molecular waste from the brain. And the paravenous efflux, like the lymphatics, eventually goes into the veins and then enters back into the body through the draining veins of the brain area, the ventricular space that then goes into the veins of the neck. It was shown through microspheres, and these are different colored microspheres, and where do they go? And the different microspheres that you have with a time lapse showed that around these arteries and veins that there was lots of movement of all these sorts of microspheres that could be injected, and these were in healthy mice. And it was pretty interesting in 2013, 2011, when these experiments were being looked at, that this was a kind of a phenomenon that people hadn't really thought about. They hadn't really thought of the implications of how things got into the brain, how things got out of the brain, and in particular, how they moved through the brain. If they were going to be, if that was a function of any particular inflammatory mediated cytokine, things of that sort. So the principal features are that there's peri-artery flow of cerebral body fluid. It enters the interstitial space through the arteriole over here. It is assisted by crossing the layer of glial end feet, assisted by the presence of aquaporin 4. Aquaporin 4 is probably a question of the boards, mainly because aquaporin 4 is pretty important. But it would be a question here because aquaporin is the precise target for the chairman of physiology and biophysics. And there's aquaporin originally had a Nobel Prize for finding it. It's a water channel. And it's the end foot of the membrane facing the perivascular space. The fluid then flows through interstitial spaces. So it's an active process. This aquaporin is open and closed and does all sorts of regulatory properties. It flows through the interstitial spaces, propelling solutes toward the perivenous conduits, leading to outflow of the brain. Remember, the artery has a higher pressure than the vein, and that the flow in the perivascular space, the blood vessels in the same direction as the blood flow, but the orientations vary. And then one of the features that tends to pump this is pulsating blood flow. And that was shown in a series of fairly elegant studies in living mice and direct observations of these things. So, metabolic waste clearance. There you go. Comes from the artery, and it sort of takes care of whatever is out there. And you've got these astrocytes, you've got neurons, you've got this aquaporin on the end plates of the astrocytes, and you have the cerebrospinal fluid. Now, certainly, fluid is not only aquaporin 4, but that controls the kind of the velocity of how this thing gets cleared. And so it comes in through the artery and then goes out by through the vein. So sleep, and the original descriptions was sleep and anesthesia facilitate trash. So awake mice, hardly any tracers enter the brain until immediately after sleep onset or induction of anesthesia, and significant tracer influx occurred. And then furthermore, sleep and anesthesia associated with a volume increase in extracellular space, which the authors suggested might reduce resistance. Now, furthermore, you say, well, if there's a volume increase and the brain is in a fixed space, what gets smaller, it turns out to be neurons and astrocytes. It increases the glymphatic flow by increasing interstitial space. Wake, you have reduced interstitial space, which restricts cerebrospinal fluid flow. In sleep, aquaporin 4 mediated changes increase interstitial space to increase CSF access, and increased convective flow augments metabolic clearance. There's color images of this whole thing that are available. And you have about 60% more interstitial space. Now, what's the analogy here? The analogy is, how do you get garbage out of New York City? Do you do it at 10 o'clock in the morning, or do you do it at 4 o'clock? And you realize that at the time in which you get things out are times in which there's not much activity going on. And the activities of neurons and astrocytes during wakefulness are such, and the development of waste products during wakefulness increases the size of the astrocytes and the neurons, and that's really what gives when you start taking out the trash. So in young people, which is the top, CFS travels through along periartery grooves, entering the brain parenchyma, and then it washes it out. In older people, the loss of aquaporin 4 water channels. Now, it's been shown first in animals, and then they go and look at the brains of humans, look for aquaporin density and channel density. They found that the channel resulted in reduced glymphatic clearance. So that helps that is shown and occurs in otherwise healthy older mammals. But in things like Alzheimer's disease, in those with Alzheimer's disease, and animals with Alzheimer's disease, or at least models of Alzheimer's disease, the accumulation of amyloid beta impairs fluid movement within the interstitial space, decreasing lymphatic clearance. So what does that mean? Well, that means that there's a target for trying to get rid of this amyloid, and that the idea of the association of the disease itself, established Alzheimer's, and impaired clearance is a target, and there are some articles on that we talked about. But you notice that this is 2020, and that you're going to see more and more. And I'm sure that there's going to be human disease presented in the sleeping. In obstructive sleep apnea, this occurs. This was just that is the clearance of the interstitial space, which can be measured using MRI, is altered and correlates with the changes in with the Epworth Sleepiness Scale, with the saturation level, with the change in saturation level that occurs with sleep, and with the AHI. Now, there's a lot of variation here, but you can sort of see there's an interaction between age, disease, length of disease, and then this particular phenomenon. And you can imagine that there are other sorts of things that are taking place, but that finding this association is one bit of evidence towards its potential role in sleep apnea. Hopefully, we'll hear something about the effect of therapy. So the idea is that there are studies that show that obesity itself, perhaps because of peripheral inflammation, that the inflammation then finds its way into the brain, decreased sleep quality, sleep apnea, reduced slow wave sleep. Here again, slow wave sleep rears its ugly head, reduced activity, and cardiovascular disorders that affect the vessels, that affect the flow, reduced respiratory breathing patterns, which are also shown to wash out things in the brain, reduced glymphatic function, or increases in proteins, metabolites, and glutamate, and toxicity that occurs and that leads to neurodegeneration. So this is a theory paper, but sort of says that every time, at least to me, that if you see a person that comes in with a diagnosis of Alzheimer's or neurocognitive decline, that when I think of them and look at them, I say, well, what sorts of things might I expect as part of sleep to impair or increase the rate of decline of their illness, or how can I affect the rate of decline? And is this program of lymphatics one of the features in this presentation? So in summary, the physiology is more effective removal of solutes and proteins in sleep than wakefulness. Wake has cellular swelling from activity, and the role of astrocytes in activity changes with sleep. Here are some articles that sort of plant a flag on the importance of this particular system in pathophysiology, MSA, Parkinson's, Alzheimer's, stroke, brain trauma. And he actually moved. And there is a literature now on depression and anxiety being a brain inflammatory state. Interesting. Any questions about part one, lymphatics?
Hey, Kingman, is aquaporin 4 brain specific?
Well, it can be found in other places, but it's particularly positioned on astrocytes. It has a very distinct function. It's also in the kidney. Aquaporins are probably best known for their activation profiles in the kidney in terms of moving water.
Got it.
On the other hand, Walter Boron has now shown that aquaporins also are affected by CO2, and he has some experimental evidence that it might be affected by oxygen. And what those two features are is that they are cofactors in how the channel works in terms of keeping it open or closing.
Mm-hmm.
So,
yep.
And they're novel targets, right? And because you have something called aquaporin 4, and you can target it or you could think about it as being important here. The drug companies, once they have a target, they're just going nuts. All right. So I'm going to now turn to synaptic resetting, sleep function, hallucinating, and enigma. How about that? Right. So, why do we sleep now in humans? And there are on the left 6 major theories that I list. It restores performance, it tunes host defense mechanisms, it conserves caloric expenditures, replenishes brain energy stores, remove toxic byproducts by glymphatic. And then the 5th is serves as a connectivity plasticity function. So the evidence for restoring performance is experimentally demonstrated as obvious evolutionary value, but the mechanism in terms of how it has that value is elusive in terms of trying to sort that out. There's really not been an ability to destroy sleep and see what an animal does in the wild, but we have plenty of evidence that when you start to destroy sleep, you start to show declines in function, which is one of the major points of that historical article in Chess. That was if you have poor sleep and you have poor health, you're hosed. Okay. B, tunes host defense mechanisms. Sure, lack of sleep blunts immune responses. Okay. Conserves caloric energy, maybe, but no evidence for clinical import. Replenishes brain energy stores. There's some amount of evidence. You can show that the energy levels are higher in the brain. You have more stored glycogen and things of that sort. That was the thing in the 80s. Removes toxic products, good support, translational, but would have to be done anyway. Connectivity changes with sleep. I like this stuff because sleep loss and changing after input are linked to sleep regulatory mechanisms. So I'm going to show you why this connectivity plasticity function of sleep is pretty interesting. So what's happening now in your brain? What is happening right now in your brain? Well, you sort of sit there and say, should I pay attention to this lecture? Should I listen to this? Should I wonder what's going on? If you're listening to something, or you're trying to learn something, or you're looking at what they call prairie dogging in your computer, to be able to jump down and jump up and see what's going on. Your activity driven changes in regulatory and neuronal connectivity is happening all the time. It's estimated that if you're driving a car, you're making about 1,000 observations a minute on how you should drive your car. And that if you're older and you only make 400 observations a minute, you are an impaired driver. And that can occur through either sleep loss, brain function, distraction, all those sorts of things that go on. So that's really the sort of the function of what you're doing. And the networks, though, are not just the cells, but the networks all exhibit a principle of what's called activity-driven changes, as in small neuronal networks. And so you have this glial cell, you have these neurons, you have synaptic terminals, you have postsynaptic neurons, you have ATP that gets released, you have neurotransmitters that get released, you have glutamate here, you have adenosine, talked about that and the importance of sleep. And then you have other things like TNF, IL-1 beta that that influence the postsynaptic neuronal pool. So remember, there's a synaptic terminal and there's a postsynaptic. And these have to sort of work well in terms of individual cell-to-cell communication, but they also have to work well in a community of cells to be able to do that. And so the thought is that sleep downscales. Does it do it all the time or not? Well, that's the truth. So what are the mechanisms of up and down scaling? On the left is is sort of a diagram, and in the box are all the things that people know about connectivity. There's the Hebbian way that things learn, and then there's scaling. Hebbian means that there's an input-output neuron on synapses, and the heavy end is the learning through other forces. Scaling is the number of synapses that occur, it's the engagement of systems. And that there's plasticity, sleep, regulatory substances, and two of them that have been well studied are TNF and neurotrophins. So wakefulness is an output, it goes in there, it works on things, and then sleep is another output. Now, the input into this thing is environmental and internal stimuli. So during wakefulness, the major input to your system is through environmental engagement, through light, sound, through those particular inputs that you react to, as well as some internal stimuli in terms of thinking and emotion and memory. And during sleep, some of these happen. So the idea is that you have something happen, it gives you adenosine triphosphates release, action potentials, glial cells. This ATP binds to purine and type 2 receptors and glia, processing and release of multiple substances, including neurotrophins, tumor necrosis factor, and brain-derived neurotrophic factors. These substances in turn alter neurotransmitter receptor populations and their location within the cell. And these actions change synaptic efficacy and the input-output state, and sleep and wake differ in their effects on the scaling of activity. And then I'm going to show the third half that they may also do it in a heavy way as well. So this is, I think, a pretty slide. And if you look on the left, what you have is this brain loop. That is your environment as you awake, sampling and potentiating your synapses to be able to understand what's going on. During interaction with the environment, the grand loop samples a limited number of inputs dictated by current events. Bottom, during sleep, the brain is disconnected through both motor disconnection and sensory, so you're not making any movements that activate your brain, and you're not seeing anything that is activating the brain. And so permits comprehensive sampling of the internal knowledge of the brain and the environment, including old memories, etc. So there's low levels of neurotransmitters combined with synchronous on-off firing patterns during non-REM sleep, such as slow wave spindles, sharp wave ripples, of conductive to synaptic down selection. So every time you see a spindle, you're going, oh, they're downscaling. It's not, oh, that's a spindle. No, I think you need to change and sit there and say, huh, that brain's downscaling. So if it doesn't have a lot of spindles, it's not downscaling, it's schizophrenia. If it has a lot of spindles, maybe helped by benzodiazepines, huh, what does that mean? So I think we need to think when we see these things happen. So synapses belong to the fittest circuits, and those that are strengthened repeatedly during wake and are better integrated with older memories and protected to survive. In contrast, synapses belong in circuits that are regularly activated during wake and are less fit with old memories and progressively depressed, eventually eliminated. So the idea of how you get rid of post-traumatic stress disorder, one of the interventions that people are now starting to use routinely in that group is to try to get people to sleep for 3 to 4 days after that event. And it seems to improve the long term. So sleep optimizes the consolidation of newly acquired information depending on the specific conditions. Consolidation during sleep promotes qualitative and quantitative changes in memory. Specific patterns of neuromodulatory activity are support system, that is brain resting state, and synaptic consolidation among groups. And slow waves, slow oscillation, spindles and ripples with minimum cholinergic activity coordinate the reactivation, redistribution of hippocampal dependent memories to the cortical slides. During REM sleep, local increases in plasticity related to immediate early gene activity, high cholinergic, and fade activity might favor the subsequent synaptic consolidation event. So this is 2010. And the clinical actionable items for these things remain to be determined. I mentioned post-traumatic stress disorder, and people have said, well, if you keep people awake, it really doesn't happen, because they just replay the memories. They replay what happened. So sleep, and so if you put them to sleep for a couple of days, it seems to be improving. So why do we study fruit flies? Well, sleep, at least in fruit flies, they have this thing called the mushroom body, which you can look at, and is dopaminergically important for olfactory memories, and it modulates sleep. And it's really interesting that studies suggest that it plays a role in synaptic scaling and calibrating the strength of dopaminergic reinforcement signals. So how does a fruit fly remember to go to a banana? How does a bee remember to go to those things? Well, they have to be able to do that. So this is learning in the human, and on the left, A, B, and C are the motor skill tasks, and on the right is a visual skill task. They are different experimental paradigms. But the one on the left, which is a motor task that you learn how to do, and this particular task is a finger tapping test, task one. And the motor is that they spend the next day awake, and then they are able to sleep. And the other is they sleep the first night, and they're able to sleep the second. What you see with the red bar there is the improvement in their particular motor skill occurs after they have a fairly good night's sleep. The visual is, and that correlates with, it turns out to be the percentage of stage 2 non-REM sleep in the last quartile of the night, which gives me comfort because I don't really generate a lot of stage 3 sleeping. So I'm glad it's not stage 3. It's at least stage 2. Maybe I could learn motors. So on the right, though, is the visual task skill. And what they do is they alternate and retest it at different times, wake versus sleep, rest of the same day, and then there's sleep versus wake. But looking at F, which is the correlation between performance and sleep stage correlation, overnight improvement is correlated with the percent of slow wave sleep in the first quarter of the night and REM sleep in the last quarter. So now we have this idea that we have these things happening in an individual when they're asleep, and that they occur throughout the first half and the last half of the night, assuming that they have this sort of consolidated sleep cycle, which in the recent chest paper said it was also the importance of sleep was to be consolidated in Arabic medicine, but not in 2 centuries ago in European medicine, which 2 sleeps were happening. So they said, okay, we've got these 2 parts of sleep, and we have non-REM and REM, and we have these sleep stage progressions, and I can take a group going through these progressions and be able to correlate their memories with their amount of slow wave sleep at the beginning and their amount of REM sleep at the end. Now, that kind of reminds you of what normal sleep has a predominance of non-REM sleep in the first half, and has a predominance of REM sleep in the second half. So it's interesting to think about then whether we could identify people using just the hypnogram of those that would have poor memory. How about that? I'm just trying to get you to look at these PSGs a little different. Okay. So this is that depth, and this is this slow wave sleep, and this is the depth of slow wave sleep. It's age dependent, and you know, 5-6 years ago, people would dismiss this slow wave sleep difference based upon the amplitude being just an artifact, but it's in animal studies it's been shown that may be important. And the age and sleep reports show that there are differences among individuals, and what on the left, the dotted lines are the standard deviation for all the people that have been measured in that particular age group, 3 to 65, and the red is the 10th percentile, and the black is the 90th percentile, and you can see that there's generally a reduction in sleep duration, but that variation can occur between 5 hours and 8 hours at 65 years as a range in terms of individuals. Now, on the right is sleep efficiency. There's where you have a really wide dispersal that gets bigger and bigger and bigger as you across the population. So if your sleep efficiency is somewhere around 75%, you might be 27 years of age. Or you're 65 years of age, maybe you don't get there, in some people actually have great sleep efficiencies. So anyway, summary here. So there's wake, there's sleep. Wake as a cellular level has its costs, cellular stress, decrease in extracellular space, saturation of plasticity. You can't grow infinite. An impairment of vigilance, cognition, and learning, if you're restricted. Impairment of cognition and learning with further wakefulness may be a function of buildup of sleep, but it also may be a function of neural circuits work over time. And then sleep improves that sort of stuff. So that's the summary of the plasticity. Any particular questions at that point? Well, let's go because this is related. Part 5, ontogeny. Sleep does what? So from animal studies, from conception, there's a period of time, 3 or 4 weeks, in which these little cells are just dividing like this. And then once they get a neural cord and they get sort of an ability to kind of twitch and do things, there's a lot of hours in the day where there's a lot of inactivity in utero. And then as in the last trimester of pregnancy, there's much more time in which there's activity. And then about 8 hours a day, there can be intermittent movements. And the EEG that's been shown mostly in sheep, because they're a fairly good, large experimental animal that you can instrument a fetus, you can bring it to term with instrumentation, you can move, you can see what happens with this. It shows that at that particular, at the time of birth, there's 8 hours or so sleep that's going on. 8 hours of wakefulness. Now, wakefulness and sleep are different in a newborn. Everybody who has a newborn realizes that they're not adults. They're not even 4-year-olds. Not even 3-year-olds. They're not even 2-year-olds. But you then have this progressive decline in the number of hours of work or during the day that occur between birth and 1 year, birth 1 year and 10 years, and then a further rate of decline steeper after 10 years of age. There's a little bump up. There's a particular lower part of wakefulness That's a, this is a change here. Okay. So what's happening? Well, at the time of 1 to 15 days, REM sleep is predominant. There's a marked drop in REM during infancy to around 2 years. And then at that particular point, it's relatively stable but declining for the rest of your life. After 6 months of age, electronic patterns of REM sleep progressively is reduced and resembles about seems of adults. And the impact of sleep on mood and affect is thought to be particularly affected by REM sleep. So, is REM sleep good for you as an adult? Well, we talked about memory. We talked about mood. Is it good for you or is it bad for you? Well, if it's bad for you, we don't have any experiments that sit there and say, I'm going to take an adult animal and instead of having, say 25% of the time of sleep when I'm going to have 50% or 60% or 70%. In fact, we don't to reproduce the infantile REM sleep patterns. So you have to sort of go back and say, well, the question is REM sleep important in development in early life? So it turns out to be that most of this has been done in animals, some have been done in humans, and this is then the EEG in what you can see in postnatal day, and I want to focus really on the right-hand side rather than the left-hand side, but I want to notice that during on the right-hand side, for in postnatal day 30, 35, 40, and P45, you see this muscle activity that's going on. And that's one of the things you see in infants, and you see in rats, and you see in all of the models that people have looked at, is there's a lot of muscle activity going on. Now, it's intermittent. It's P45 is probably a slow wave equivalent. But you know, when you mention and talk about this sleep, we use adult terms, but that's not an adult. We can't ask them, did you sleep well last night? Right? We can't ask them certain questions about function because they really can't function that way. Everything is amodal. Their sensory comes in. Light, hearing, touch, it's amodal. So you sort of have, well, this is the EEG activity, this is what's going on. But people have focused not just on the EEG, but also this idea that there's motor activity. Now, why is that important? Because motor activity, twitches, particularly when they occur during REM sleep, that they are activating the brain. They are afferently going up into the brain and sort of probing that brain for what it can do. So the active inhibition of muscle tone throughout the body is not seen in animal models or as it's not been looked at carefully in humans. And people look mostly at breathing and heart rate because of implications for sin, but in terms of sleep. So on the right-hand side, what it is that if you sleep deprived rats at these different postnatal days, and postnatal day 12, 16 are kind of like premature humans. Equivalency is about postnatal day 20, and then postnatal 24 sort of is when they start to look like a 1 month or 2 month. And that's when you start to get a delta rebound sleep. Now, how do you prevent sleep in these animals? Well, you measure them, and you look at them, and you kind of gently wake them up. Is that a stress? Yeah, it probably is. But you don't change their patterns of what it looks like of delta power, slow wave sleep, until you start to get out to some particular age. So you're not able to generate a sleep deprivation rebound and slow wave sleep until a particular age of brain maturation. So neonatal REM sleep is regulated by corticotrophin releasing factor. Another one we can do. What does adult REM? Acetylcholine, right? So corticotropin releasing factor. That's different, isn't it? Cholinergic neurons have been recognized to be a major source of rapid eye movement in adulthood, but their effect on neonatal sleep remains to be discovered. It may be the CRF somehow enhances REM sleep and associated with some sort of cholinergic activation. But it's not. But it's not exactly that. And I think that's a pretty important statement that says that neonatal REM sleep is very good for adult REM sleep. So how do you get this? REM serves as an endogenous source of stimulation, furnishing great quantities of functional excitation to higher centers, both internal as well as the twitches in the external motor tract. Such stimulation would be particularly critical during periods of in utero and shortly after birth, where appreciable exogenous stimulation is available to the central nervous system. So I think when you sit there and you think about this, you imagine your baby being available to all the things that you know of. All the things like, oh, that's a car horn. No, that's a limb breaking off. Oh, no, that's a boy over there. Oh, that's a girl. That's it. They don't have any of that. They just see movement. They just see activity. They just hear things. It's amodal. Oh, that's somebody touching me that's somebody holding me and just drop me a little bit. And then you get a more response, right? I mean, that's how you do it. And that's why you do it, because you want to say, how integrated is this neurological system? The Babinski goes away, but comes back, right? All these things happen. So in slip, so assist in structural maturation and degradation. So the first studies were done by blocking monocular light into the system and interfering with REM sleep. And I want you to see what's in the box here. And this is the number of frequency of cells that are there in terms of cell size distribution. And in those particular with one eye part of the occipital lobe versus the other eye, is that there are different cell sizes. Cells are still there, but some cells are contralateral cells are missing. The larger cells are missing. Eyes come go one way or the other, and so why does light coming from your left eye, is that gated according to what REM sleep is all about? I think that's pretty interesting, and that was found in science by a guy named Harry Ruffler, who's one of the bigger. 7 days of selective REM deprivation. How'd they do that? They measured them in REM and they woke them up. Now guess what state they had, or not, but you gently got them out of this. What state? They had slow wave sleep. And you go, it's not that they can't generate slow wave sleep. It's just that they don't generate slow wave sleep unless they're interrupted from REM sleep. Okay. So there is a recovery. That is, if you unpatch them at 7 days, period of extended developmentally regulated synaptic plasticity could be elicited in slices of the visual. And they support the role of REM sleep and its associated neuronal activity and maturation. And this was in 1963 or so. This is a review in 2002. It's now been, there's a person at the University of Pennsylvania that's taken this up, a junior person, and is really looking at molecular reasons for this. Making hay to make a lot of that. So the other thing we did here is we did REM deprivation in neonatal rats between P3 and P10. We did two things. We did perinatal administration of REM suppressant, and we used several different REM suppressants, and examined then in adolescence helplessness learning and new learning for depressive traits, and found that in the adolescent animal that this was inhibited. And everyone said, well, it's the drug. So they went through and did neonatal sleep, REM sleep deprivation induced by a non-drug measure. They measured the EEG and would gently move the animal into REM sleep, and you could reduce REM sleep by almost 80%, and you created an adult depression-like changes, similar to those induced by REM sleep suppressant drug. Extent was different. So this was Peng Fu Feng over the DA from 2001 to 2014 or so. Other observations: neonatal REM maternal deprivation produces less active REM-like sleep and adult hyperarousal syndrome in adolescent rats, and that has been shown in humans. Adult MD rat could be a model of insomnia with cholinergic mechanism. So we use that as a model of insomnia in adult rats. Early life mood disorders have been linked to drugs taken during pregnancy, hospitalizations, that is being in the hospital and leaving the hospital after the mother has left the hospital. And then certainly in environmental distress and warfare. So high REM sleep varies among lots of species. It's interesting, those that have greater than 3 hours of REM sleep per day, platypus, ferret. Those that have less than an hour, guinea pig. Didn't know that, but we got a guinea pig. But sheep, horse, and giraffes. So REM sleep selectively prunes new synapses, anatomy, newly formed spines, strengthens the fraction of new spines formed during development and learning, facilitates long-term maintenance of new spines during development and learning. And there's a molecular mechanism people injury, calcium spikes, regular sleep, and motor training. So the brain ontogeny hypothesis, they show the association between amount of sleep, certain indices of brain development, neonatal REM sleep deprivation, leads to abnormal brain and behavioral development, and perinatal, adolescence, and early adulthood in critical development periods. An ontogenetic hypothesis, which you'll hear every now and then if you read the basic science literature, determine REM sleep influence development programs and activity-dependent development. And that's important because you really develop things through activity. And then more generally, the main function of sleep in adults is to scale back some. Okay. I think that's what, yeah, that's what I did. Thank you very much. And this was the presentation. It's improving, it's updated since I gave it to the graduate students. And I'd like some feedback on it.